So following up from the previous lecture, let's look a little bit in modeling heat stress, and then we will have a longer break, and then thereafter Aurélie will continue on this topic. So as I said also in the previous lecture, if you model G by E, or the environmental sensitivity of individuals relative to temperature or heat, that typically starts above a certain threshold. And for this, most applications, people have been using the temperature humidity index, which is a combination of the temperature but also the relative humidity, as I will show later. So that if we have a relative humidity of 80%, then for a temperature of 10, the temperature humidity index is 51, and it goes up to something like 83 if we have a temperature of 30. This is also something that you will be working with in the exercise this afternoon to get a better feel for those values and how this is calculated. So if we think about heat stress in dairy cattle, a number of things may happen once it gets really warm. So milk production will go down, immunity of animals will go down. There may be more disease pressure. The somatic cell count of individuals may go up, and fertility may also go down. And that's sort of looking at the output side in terms of what happens with the traits that the individuals express. But if we look more internally, what happens in the individual, well, it's likely that the body temperature is going to rise, and the respiration rate to try to get rid of this excess of heat is also going to increase. The animals may consume more water, but actually have a lower dry matter intake and lower rumination, because eating the feed and then actually transforming this into something else that also creates heat. So it may actually be a good idea for the individuals itself to reduce foraging activities. So if you want to model the impact of heat stress, then for the individuals that you're going to work with or for your data, you need to obtain both the relative humidity and also the temperature. So there are different ways of doing this. Probably the most accurate would be to measure this on farm. But the issue with this is that the availability is relatively sparse. And even if farmers would measure this on their own farms, unlocking this data or bringing this together across very many different farms may be difficult. So something that's used quite often in different studies, we also use this, is to use the nearest weather station to a particular herd. The good thing about this is that the data there measured is typically freely available, but you do require postal codes of the herds to be able to connect the weather stations to the herds. And this may be an issue if there's things related to privacy of farmer-owned data. So typically, if we use data from practice, we would get it anonymized, and if it's anonymized, you don't have the postal codes. Nearest weather stations work relatively well in densely populated areas such as the Netherlands or Northwestern Europe, for that sake. But the distance may be too large in more remote places. And that's where people have started to use meteorological data from NASA Power, which is derived from satellites, but it also includes different types of measurements to really try to predict temperature at a given moment in time and in a specific place. So as I said here, in the Netherlands, we would use information from weather stations. Here is a map of the Netherlands. Well, if you're not familiar with how big or how small our country is, I think if you go from here to the top right, that's about 400 kilometers or something like that. So distances are all relatively small. And you can see there's somewhere around 30 weather stations scattered throughout the country. So when we used this information, we found that the average distance from the dairy herds in the data to the nearest weather station was a little less than 15 kilometers. And then the largest distance was only 36 kilometers. So that's pretty good, especially in a country, well, where the climate is relatively similar. Although it does make it different on certain days if you're closer to the coast or not.
Especially if you have a flat surface.
Yes, yeah, that also makes, yes, yeah, indeed. So the Netherlands is very flat, so there's no mountains that cause differences in temperature or stuff like that as well. So based on this publicly available data, you can then make plots to look at the average daily temperature. This is in a place called the Bilt, so that's where the main weather station is, more or less in the center of the country. And then you can also see how the temperature changes throughout the year. So to compute this temperature humidity index, there's a formula here that is typically used in most studies that have, well, that in the last 10, 20 years. The formula may look a little bit complicated, but well, if you look a little bit longer to it, you will see that the temperature humidity index will increase with the temperature, and the relative humidity doesn't have too much of an effect on the outcome, which I will also show on the next slide. But it only has two inputs. It's the average daily temperature in degrees Celsius and the relative humidity. So here I plotted for the x-axis a certain temperature, and then there's three different lines for three different relative humidities, and you can see that those lines are all very closely to each other. So in most cases, depending on the variation in relative humidity, the temperature humidity is close to being sort of a linear transformation of the temperature. So to model heat stress, in different publications over the recent years, this has been done using three main steps, and I'll guide you through those. First, you need to determine what is the temperature humidity index thresholds where heat stress starts. Then you need to parameterize the heat stress at a population level, so find the overall pattern of performance as function of temperature humidity. And then the last one is to parameterize it at the individual level. And that could include a genetic effect or a breeding value, but also a permanent environmental effect. I'll talk you through some work that we've done here for conception rate in Dutch dairy cattle. So, and we wrote a paper on this that will appear, that's accepted in Journal of Dairy Science, so should appear in the next months. So what we focused here on is first insemination records in Holstein cattle between 2010 and 2022. We looked at conception rate at first insemination for a little bit more than 400,000 first parity cows. And then at the moment of this first insemination, we obtained the THI obtained from the nearest weather station. So first, what you need to do is to evaluate the effect of the THI on the average performance of all the individuals in the data. And you do this by running a relatively simple model that corrects for some systematic effects, such as herdier season. And in that model, there's a fixed effect for THI, and every discrete level of THI, so starting somewhere with values of 20. So all records that have a value of 20, they are one level. All that have a value of 21, they have another level, up into The highest value we saw was something like 78. If you do this, there may likely be some confounding. So in the summer, the temperature and humidity is going to be higher, but that's not the only thing that changes between summer and winter. So if animals are grazing, maybe that may be better for them to some extent if they're outdoors in the summer. On the other hand, availability or quality of feed may be different in different seasons. So there may be some confounding between temperature, humidity index derived effects, but also contemporary group effects. That's something to be aware of. The good thing is that if you group individuals that experience the same level of temperature humidity index, you're going to group individuals across many different herds at the same level. So you hope that the main effect due to the temperature and humidity index is really captured by that factor and not all the underlying contemporary group effects of these same individuals. So that really states that observation per level of THI are typically much higher than number of observation at a contemporary group level. So you would expect that the main signal due to the THI should also end up in that effect in the model. So we did this here for this particular data. I left out the x-axis. This is because you will have a simulated version of this data to work on this afternoon. So you have to work out yourself what those values are. But here, as you can see, so what I modeled here is the estimate of the fixed effect for different levels of THI, expressed as deviation from the population mean. And that, well, for the lower values, it sort of hovers around zero, so there's not really a deviation. But then at some point, the conception rate really starts to fall down. So based on this, you can, and you will identify this threshold yourself this afternoon that has a certain value. So that's the first step. Then you know what the threshold is. Then the second step is to parameterize the heat stress at the population level. It's a bit similar to the modeling of resilience, where you first, for individuals, you want to have the smooth curve and then the deviations of those. Here, it's what you do at the population level. You want to find the smooth curve of response at the population. So looking back to the pattern I showed on the previous slide, you just have to look at this and try to come up with a sensible curve that would fit this rather well. And the benefit of having a parameterized function is that compared to fitting each THI level as a separate class effect, is that in some cases, especially at the lower values, perhaps there's relatively few observations. But if you parameterize a function, then the estimate at a particular value of THI also borrows information of neighboring THI levels. So what we see here, up until whatever the threshold is, there's a more or less stable performance, and thereafter there's a rapid rate of decline. So the typical function to model here is, once you know what the threshold is that you do a regression on a parameter that has a value of 0 below the THI threshold, and then above the THI threshold, it's a regression on a covariate that is a function of THI. And again, this is something you will be working on this afternoon. So once that's done, you have this overall pattern. Then the deviation of each individual relative to this overall pattern can be modeled to obtain individual breeding values as function of THI. So in this sense, we would model them as some sort of a reaction norm or a random regression. And we use a similar kind of approach as for the overall curve. We do, in this case, we do a random regression, so the overall curve would be a fixed regression. And we define a covariate for the individuals that any inseminations that were at THI levels below the threshold, the covariate is going to be equal to 0. And above the THI, the threshold is proportional to the THI. So what we did in this particular case is that it was really a linear function above the threshold. It was a linear function of the THI, such that above the threshold, we basically fit linear reaction norms, if you like. And when we did this exercise, here I plot the breeding values for conception rate of the 30 bulls that had most information in the data. So they had the highest reliability for the breeding values. Up until the THI threshold, it's just a straight line, just by construction of the model. We assume that THI does not affect the breeding values, but then thereafter, also by construction of the model, breeding values are a linear function of THI, and you see that there's some re-ranking starts to happen. So just to highlight two bulls here, one indicated in green and one in red. Up until the threshold, there's basically no difference in breeding values between the two. But then beyond the threshold, at the very highest observed values of THI, we can see there's a difference of about 6% conception rate. So this suggests that modeling this properly and taking this into consideration could help to start selecting bulls that actually give daughters whose fertility at under heat stress is better than some of the others. So this kind of model in the literature has been termed a broken stick or broken line model. And that's really because of the nature, how the model is constructed. And the first pages on this already date back to the year 2000. So here the idea is that you have, when we typically use THI on the x-axis, is that the trait value is not affected by that up until the threshold, but beyond the threshold, there's an overall effect that the trait value goes down, and then the degree to which animals are affected may differ between them. Another thing that they suggest in this paper is that it could also be that the threshold for individuals may be different. But I don't really think that anyone pursued this to try and estimate this in literature, because the modeling simply becomes increasingly more difficult to do this. Okay.
He tried to do that, but it's almost impossible.
Right.
If you don't have information in that.
Right. Yeah. So just to summarize this part, modeling heat stress is typically done in 3 steps. First, you need to determine the THI threshold where heat stress starts. Then you need to parameterize the heat stress at the population level, so the average heat stress in the population, and then the deviation of that at the individual level, that's the third step, gives the breeding values of these individuals. Questions about this?
Do you use a sign or an animal model?
That's a very good question. So in this particular study, where I showed these results, we used the SIRE model, and I think maybe we start initially with an animal model, but the problem is that this is a trait where each individual has only one phenotype, right? So you want to model the breeding value of the individuals as a curve, but you only have one observation to parameterize the curve. So if you run a SIRE model and you do it based on daughter information, these sires will have multiple daughters with information, and therefore the observations that pertain to a sire, they will be scattered probably along the THI axis, and really unable to estimate those curves. It also makes for a much sparser model and easier to estimate.
And you estimate what is the effect of not fulfilling the conditions to run a SIRE model?
I'm not sure what you mean with that.
Models are equivalent given number of conditions.
Right.
So what is the effect of using a SIA model when you clearly know that those conditions are not fulfilled in this data set?
Oh, no, we did not. No, we didn't do that.
Because we have the same problem, we have estimated the heat stress on winning wave, fertility rates, and we have the same problem, just you had just one observation there.
Right. Yeah.
So basically you rely on the pedigree and genomic information.
Yes. Yeah. And then, so you hope that to parameterize the curve of an individual, you can leverage the phenotypes of related individuals that have experienced a different level of DHI.
Basically.
That's the theory, but then in practice, it's.
Because I was thinking of using the SIA model, though it's a little bit more complex because then you have the maternal component, which is not that easy.
Right. Yes.
So
Yes.
it's a little bit more complex, but I was wondering if somebody knows what is the effect of that before we go for that.
No, I don't know. No.
Because we tend to forget that. I mean, we tend to assume that SIA and models are equivalent and that they are not. That's the reason why we went from a SIA to a random model.
Right. Yeah. Yes. Yeah. I'm not really sure if it would be easy to model this in terms of resilience because you only have like a single observation of an individual, right? So there's not a longitudinal pattern to try and derive this from. So.
There is a way to make the theoretical calculus that anymore. The fact that you expect to complete. But then the variance, you can make it so that animals that are able to cope with the perturbation, right?
Yeah.
Perform at least the same or better, right? Then your variance will still indicate animals that are disturbed by the perturbation. There may be a solution in the way you actually set what's the ideal performance of the undisturbed performance.
Yes. Yeah, but I'm not sure how accurately you could try to predict that. No. So that could, yeah.
It's a resilient rate on the science side of it. But you have longitudinal data.
Yeah, true. Yeah. You could model it at the level of the sire. Yes. Yeah. Well, in a way, this is maybe it would be similar to what we try to do here. I think so. Yes.
Instead of having conception rate, you have the reproduction of factors, whatever. You have a serial model because you have longitudinal data.
Right. Yeah. And the other thing, I said that we use THI here measured at the day of insemination. Actually, we took the average of a 3-day interval, including 1 day and 1 day before and 1 day after the day of insemination. But we also tried increasingly longer intervals surrounding it, and we found that the effect that we see of heat stress, like the longer you make this interval, the smaller the observed or the estimated effect. So it really seems to be on that moment is what's most important. So in this, it's really sort of, you could see it as a short-term perturbation of that the animal experiences. Yeah. Okay, if there's nothing else, then sorry.
You were on total 50 points or you choose those randomly?
No, those are the ones that had the highest reliability of the breeding values. So these should have had the most daughters throughout. Actually, I looked at the standard errors of the slow breeding values of these individuals for this part, and I took the ones with the lowest standard error. So they should have the most information to estimate these breeding values.
And what is the interpretation of that?
Of what?
Of the slope? Because it's not that I mean, you see the figure like this, and you see at high PHI, the cow is better than.
Yes, and that's not the case. That's not the case, right? So these breeding values, they are a deviation of the overall curve, right? So, and the overall curve is something like this. So if we pass the threshold, on average, the daughters of all bulls will perform worse. But then if that breeding value shows a positive trend, it just means that the daughters of that bull are not as much affected by the higher temperature than daughters of some other bulls. So that's a very good point. Yes. Yeah.
There's no correlation between the breeding value without the slope and the slope direction. They seem quite scattered around.
We did estimate them, but I don't know them from the top of my head, to be honest. They're a bit scattered, but it's, the ones that are poor here, they don't become the best ones there, vice versa, right? So they do, within a certain bandwidth of the breeding value, they do re-rank, but the best one here is still the best one regardless of the DHI, right? And the worst one is also almost the worst one regardless of the DHI. So I think there's positive correlation between the slope of this and the level that they are. No, of that. No, but if you look at the breeding value at the highest DHI level versus the ones below the threshold, there's going to be fairly strong positive correlation between that. So that if we ignore all the complexity, and basically this is the breeding values we would get in the models that we use today, we would still be doing something that's also relevant at higher DHI levels. Okay.
No more questions? No? So thank you, Marion, for these two presentations. Now we'll have a break until 11. And then we'll start with already to present also the results on that. So thank you.